So the title of Chapter 15 is Genomics and Proteomics. The Human Genome Project and other genome projects that have been completed or are being worked on fall under the category of what we call structural genomics. And structural genomics aims to sequence the entire genome of an organism in order to study it. And the basic approach of any structural genomics or genome project would be to map a genome, marking where important markers and genes are, and that requires both what we call physical maps and genetic maps. So you're familiar with a genetic map because that's a map based on recombination frequencies. Then you have to break the chromosomes into pieces, into fragments, and clone them into vectors, so make a library, and then sequence the entire library, sequence the DNA in each of the clones, and then use a computer to help you reassemble all the fragment sequences back into composite chromosome nucleotide sequences. The Human Genome Project in particular began in 1990, funded by the NIH and the DOE, Department of Energy. So it's all tax money public money that funded the project. In fact, most research in the United States is funded by tax money. And the Department of Energy was involved because of its involvement in the Manhattan Project. It was interested in studying DNA and mutation rates, and therefore has always been interested in studying mutation rates in DNA. And so that's how that got tied in. The project was headed by Francis Collins, shown here. He's an MD, PhD, and the initial project was supposed to be completed in 2005. So they had a 15-year timeline for this. And it cost about $3 billion. Now, shown here in this picture also is J. Craig Venter. And he was, at the time, the CEO of a company called Celera. And in 1998, it was a private company, they announced that they intended to sequence the human genome within 3 years, and they had their own private money to do so. Now, by 1998, the Human Genome Project, which, by the way, was an international project that had many countries that working on it, primarily the United States taking the lead, but other countries that were helping with that. And so they had been working already for 8 years doing the mapping and initial sequencing. So when Celera Corporation announced that they were going to do the same thing, it's a direct competition. And the Celera Corp did not promise clean, accurate sequence, but just intended to do what we call a dirty sequence very fast so they could patent all the genes they could find. Now, in 1998, the scientists of the Human Genome Project, including myself, recognized that this might be possible, and so it was a direct competition. The interesting thing, though, was that the Human Genome Project was aimed at sequencing the chromosome sequences almost perfect. We were only allowed to have one error or one uncertain nucleotide for every 10,000 bases in a sequence. So 1 out of 10,000 was our error rate. And Celera did not promise any such thing. In fact, when Venter talked to Collins, he said, "We'll take over the human genome. Why don't you guys just sequence the mouse genome instead?" So he was very arrogant. Well, the Human Genome Project responded to the challenge and shut down all of their centers except for the 3 largest ones. And I worked at UT Southwestern at the time, and we were one of the smaller centers. So we were shut down, but the 3 largest sequencing centers began to do a quick and dirty sequence of their own, because the Human Genome Project sequence data was always put into GenBank. GenBank was that database that y'all typed your DNA sequence into in lab, and you did a blast comparison analysis. And so that GenBank database is publicly available. And so the human genome sequence data had always been uploaded to GenBank and still is. And so if the Human Genome Project could finish a quick and dirty sequence, not a perfect sequence, but a quick and dirty sequence fast, and put the data up on the GenBank database, then once it's publicly available, Celera would not be able to patent any of the genes, because you can't patent something that's publicly available. Well, in 2000, it was announced that both groups had completed a draft, and so I say it was a tie. So neither group really won the challenge. A draft of the genome doesn't mean that the pieces had been assembled into all the chromosomes, but just that all of the clones from the genomic library had been sequenced. So it was still up to the computers and the scientists to put the sequences together. In fact, the Celera group admitted that they had to use the publicly available maps that had been published by the Human Genome Project in order to work out their assembly of all these sequences. And both groups published an initial analysis of the human DNA sequence, human genome sequence, in 2001. In 2003, they declared that the sequence was finished with only one error per 100,000 bases, and there were still a few gaps in the sequence, but still pretty good, and 2 years ahead of schedule. So in some ways, the challenge by the private company probably sped up the progress of the public genome project. One thing that we found from the Human Genome Project was that when you sequence chromosomal DNA from different individuals, you find most of the sequence is exactly the same, identical between two individuals. But there are certain locations in everyone that tend to be variable, and those have been named SNPs, or single nucleotide polymorphisms. So here you see 4 individuals whose DNA has been lined up in this one area. And so you have individual A, individual B, individual C, individual D. And you see on this chromosome, individual A has a C at this position, and some individuals have a T, for example, so that would be an SNP where if you lined up hundreds of individuals, there would be some variation at that position. Here at this middle one, this middle SNP varies between a G and an A, and this one over here varies between a G and an A, and so on. So you have all these where most of the nucleotide sequences are identical, but you have certain locations that tend to vary more commonly. And so those are called SNPs. And then what's really interesting is that certain people will have a group of certain SNPs, and they call those a haplotype. So individual A has a C at the first position, a G, and a G. That combination of nucleotides at those 3 positions, that grouping is called a haplotype. And then you have individual B has a C and an A at the 3 positions, and so on. So certain haplotypes are a group of different, these are really alleles for each SNP. And certain haplotype makeups have been we start to correspond certain haplotypes to certain predispositions to diseases or predispositions to reactions to pharmaceutical drugs, and so on. So this is just sort of the in-depth analysis that we are able to do that started with the data from the Human Genome Project. And so now you have a map of all the haplotypes or all the SNPs, and that's called the HapMap.